Yesterday we were talking about the expected value, the E value of match run, and this match run between two sequences of length m and n. These are the possible places we could have a match. Is basically this whole matrix. We can start anywhere. If you are talking about single matches, the probability of observing a single match, it can be anywhere in this matrix, and this matrix is of size m times n if one of the sequences of length n and the other is of length m. And when we're talking about longer matches, longer matching runs, then what we do is we cannot use all parts of this matrix to start the matching run. For example, when you're interested in a match run of length 3, it can start, we should look for diagonals of length 3. For example, this is of diagonal length 3, but we cannot start a match run at this point in this matrix because we are out of letters, we are out of amino acids, so we can at most have two matching runs. So we cannot use all the m times n positions to start a matching run. However, this can be ignored if the length of the match run you're looking for is much smaller than the actual length of the sequences. Okay, if m and n are like 100, 500, and if you're looking for a length of 10, this can be ignored. So we can approximately say that, for example, the length of the expected number of two successive matches is going to be 10 times 8. 10 times 8 is the size of this matrix, and this is the probability of observing a single match run of length 2. So similarly, we can use therefore you can use the same equation. So if we have two sequences A and B, and if the probability of occurrence for every symbol is P, then a match of a residue AI with BJ is P. Okay? If for example, if you have 20 amino acids, the probability of matching is 1 over 20. The probability of having two specific amino acids at the same position is 1 over P square. But we are not interested in specific amino acids. We are just saying match. Okay, we are not just saying match of two alanines or match of two glycines. They are both matches. So therefore a match of length L from AI BJ to AI plus L minus 1 to BJ plus L minus 1. As you can see, we do not have gaps here in this matching run. We are not analyzing matches with gaps. We do not have gaps. And all matches, no mismatches. And that probability is P to the L. To observe 10 successive amino acid matches, for example, is going to be 1 over 20 to the power 10. Okay? So the coin toss problem corresponds to the longest run of matches along the diagonals. Okay, so this keeps me, okay. And then we can use the same formula. If there are actually this many places, m minus L plus 1 times n minus L plus 1 places where the match could start. So this is the actual number of experiments that we can try this match run. But if L is smaller considered with respect to m and n, this can be ignored, and we can directly write similar to the previous formula of n times p to the L. We can say m times n p to the L. And the expected length of the longest match can be approximated as this formula, which is very similar to the Hedron formula. Instead of n, we have m times n. And here m and n are the length of the two sequences. And here's an example. If we have two DNA sequences of length 10, okay, they are not very long. And if in your alignment you see that there's a match of DNA sequences of length 3. There's an exact match of 3 nucleotides between these 10 DNA sequences of length 10. And this is something actually expected, okay? This is, you get any 2 random DNA sequences of length 10, there can be, the expected number of match of length 3 is 1, okay? So this is not surprising. So if finding an exact match, if you are looking for example, BLAST was looking for exact matches between sequences, it means that any random sequences could get an exact match of length 3 if your DNA sequences was of length 10. So this is not surprising because we only have 4 letters in DNA. If you do the same analysis, if they were protein sequences, the expected length here would be much smaller. And we have certain assumptions when we do this. Base composition means that we assume that A, C, G, and T are equally probable. They are all 1 over 4. Their probability of occurrence is 1 over 4. And we do not consider any gaps. But still, even with these assumptions, this is a good estimate. And now we are going to, up to now we have been talking about length, okay? The length of a matching run. And, but do not forget that we use scoring matrices. Usually at the end what we are going to be given is that, when we are given an alignment, we are going to be given an alignment score. And we'll be asking how significant is this score? Is this score, can we get the same score or better score by chance? So at some point we need to extend this idea of length of matching runs to scores. If all the matches were scored equally, if all the matches were scored 1, okay, then this would be exact, the extension would be trivial. The length of the matching run would be the score of that local alignment. Now we are going to see we can actually work this equation if the match scores are something other than 1, and even if it changes for every amino acid, we have a certain scoring matrix which defines different scores for different matches. We can still come up with a similar formula to tell us the significance, the expected value of a local alignment without gaps with certain score. And here, and also if we consider all the mismatches receive a negative score of -infinity, and this AIBJ match receives a positive score of a certain score that we take from a scoring matrix, Sij, we can estimate the expected number of matching runs with a score X or higher is proportional to mn p to the x. So this formula says that we can directly replace the length with a score because it's going to be proportional, but it's not going to be to get the exact expected number. We need to work out how the scoring matrices are built in the first place. What do they mean? What does a score of 7 means? What does a score of 3 means? So we can accommodate for this. At the end, our goal is to convert this 3 to 1 match. Okay, it means that the score of 3 between these 2 amino acid matches means it's a single match. To understand it, to accommodate for this score, in order to convert the score to a length, we need to work on the details of how the scoring matrices are built. Okay, so Carlin and Altschul actually developed this theory. Okay, the theory for statistics of local alignments without gaps, and also firstly the theory was only using a match run. It didn't allow any mismatches. Then later it was also extended to allow for mismatches. We could have negative scores between certain amino acids as well. So here is how it goes. So now let's talk about statistics of local alignments without gaps. What we need to have is, in order to have a meaningful e-value, our scoring matrix, that the scoring matrix we use, which scores matches and mismatches differently, has to satisfy certain constraints. And that constraint should be, the main constraint is the expected score of a single match obtained by a scoring matrix should be negative. It means this: just pick randomly two amino acids, just pick two amino acids randomly, and what is the expected value of this their match, okay? Match or mismatch, okay? You just pick two amino acids randomly. They can be different, the 19/20 probability they are going to be different amino acids, okay? Because they are the same amino acids. The match probability is 1/20, okay? And 19/20 probability they are going to be different amino acids, and the expected value of this. So you just pick two random amino acids. What is the score of their match or mismatch? The expected value should be a negative value, okay? This means that if it wasn't negative, otherwise, if it wasn't negative, it means that by just having a random alignment of very long length, by just taking two random sequences which are very long, you could obtain a very high score by just randomly aligning. If you randomly align these amino acids, if your expected score of a random amino acid match is positive, it means that by adding these positive numbers infinitely many times, if you have a very, very long sequence, you can expect a very high score. So therefore, obtaining a good score could be between random sequences of between very long sequences. Therefore, in order to have a meaningful e-value, our scoring matrix should satisfy this, and this e-value can be computed by this. It's basically this. So this Pi and Pj here are the probability of occurrence of a specific amino acid. If in nature they do not occur in equal percentages. If they were occurring in equal percentages, the PIs, all the PIs would be 1/20. Okay, I is equal to I is between 1 and 20. Okay, if all the amino acids in nature, so this Pi here is the probability of observing an amino acid I, and this is Pj is the probability of observing an amino acid J. If all of them were equal, we would have these would be 1/20 probabilities, and this is the score of aligning I with a J. So it's like this. We have 400 different possible pairings of amino acids, right? In these matches, we have these 20 amino acids, A, C, D, E, and it can be matched to any of these other. But if we consider the symmetric, it's not going to be 400. It's going to be 200. If we consider the symmetry, the matching of A with a C is the same of matching C with an E, then it would be symmetric. But they're going to, what we do here is, this is just a weighted average of all the scores should be less than 0, meaning that if you just randomly pick any amino acids, the expected value of their score should be negative. And this is what will happen otherwise. This is what I just stated. And if this requirement is met, then the expected number of alignments with score X or higher can be given by, so in this previous formula, we just stated that it is proportional to the score. The e-value is proportional to m times n, p to the score. Now we can actually give an exact formula, state that what is the expected value of to obtain an alignment of score X or higher, can be given by k times mn e to the minus lambda x. Okay, so here there are two new constants you see here that we haven't mentioned earlier. So x here is our score, our alignment score is x. And what we have here is we have lambda. We multiply the score x with lambda. This is just to convert the score to a length, okay? Because the exact formula here, we have actually what we have here is p to the l, and here we converted this p to e. This is what is different, and this x, the score of x is multiplied with lambda. So this lambda actually is going to be a constant that we are going to compute, which is specific to a given scoring matrix. So Blossom 62 is going to have a certain lambda value. It is, or if you multiply the score you get with this lambda, you convert your score to a length that you can use. And k here is just a constant to account for the fact that we cannot start this matching in every matrix position. So that's the only, remember we were using this m and n directly, and this expected value was something approximate. Then this k is just a constant which accounts for the fact that we cannot start the alignment everywhere in the sequences. At the ends of at the boundaries of the sequences, an alignment of length, a certain length cannot start at the end because we ran out of sequences at the ends. This is k is on, yeah. This slide describes what they are. Okay, it's something less than 1. It's a proportionality constant that corrects the mn space factor for the fact that there are not really m times n independent places that could have produced score s greater than x. And k has little effect on the statistical significance of a similarity score. It's a constant. But lambda is really important. It's found from the scoring matrix that is used. It's specific to the Blossom matrix or PEM matrix. It has different lambda values. And it takes into account that scoring matrices do not contain actual probabilities of co-occurrence. Okay, the scoring matrices, if you remember, they just contain integers, right? If you try to remember the scoring matrices we use, the match scores, mismatch scores like 7, 10, 11, minus 3, they were not probabilities. So it wasn't, but instead they have a scaled version of these values. So in order to actually compute lambda, we need to look how these 7, 11, these integers, how are they computed in the first place in a scoring matrix. So let's look at the details of computation of scoring matrices. The first scoring matrices were built like about 40 years ago by using manual alignments of sequences. So this is a multiple alignment of a family of sequences. What you see here is a portion of a family of protein sequences which are aligned, meaning that related amino acids are put under the same column. So the same column represents the same amino acid, meaning that, for example, this L, when we see this, it's changed to YY in the first two protein sequences, means that there must be a mutation. There was a mutation which in these species, in which this L is changed to a Y. Okay, so these alignments are done manually, and as you can see, sometimes there are gaps, very few gaps, but this alignment was done manually. And the main purpose of having this alignment is that we want to find out which amino acids can be mutated to which other amino acids. So meaning that in this alignment, we see L against the Y means that if you're trying to align completely two new sequences, aligning L with a Y should be preferable compared to aligning L with an M, for example. Because in real sequences, we see L can be mutated to Y. So when you're doing the alignment, of course, at the first place, you try to align same amino acids. You need to align, you want to maximize the score in your alignment, you try to align L with an L. But if there's no L in the neighborhood, and imagine there are two amino acids, one of them is Y, the other is M, you should prefer Y to M, because in nature, L and Y are related. One of them is mutated to the other, and still the protein has the same function. If we have this mutation, this is an observed mutation in nature. And so when we are doing the alignment with considering these actual observations, we would get an alignment which mimics nature more. Okay, that's the main purpose. So then here, what they want to find out is that how likely two different amino acid types appear at the same column. Okay, so here what they do is you count how many, for example, there are this many amino acids here, how many possible pairs we could have, and the expected number. If we do not know anything, okay, actually it's 1/20. So if we do not know anything about this, if we do not have the alignment, to the probability that given a specific amino acid A, if I just pick a random amino acid, the probability that I'm going to observe these two together in the column is 1/20. Now, what we do is, by counting the actual occurrences of these amino acids in these alignments in the same column, so we are going to count within the same column which amino acids occur together. And we are going to, by comparing that number with this 1/20, we are going to see that whether it is more likely than random to see these two amino acids together. So this is called the likelihood ratio. Okay, so by random chance, we expect them to be in 1/20 probability to appear in the same column. But if you look at the exact frequency in this alignment, if it is 4/20, it means that it's 4 times more likely that these two amino acids are together compared to random. So this is what they try to do, mutation likelihood, how likely to mutate to a certain amino acids. And here is what they compute is what is called the log odds ratio, okay? And the score score is computed as this log odds ratio. So what does this mean? Here, Qij is the observed frequency of co-occurrence, okay? And Pi and Pj are the individual frequencies. It's like this. If we just count, let's look at how many, so it's, for each amino acid in this alignment, we count in every sequence how many times this, so we count all the letters, okay? You just, what you see here is a block of letters, okay? Now, the first thing we need to account for is certain amino acids may appear more than other amino acids. So there may be certain amino acids which are very common. So we need to, of course, what I say here was, I said 1/20, but this assumed that all the amino acids occur at the same frequency. But obviously it's not the case. Some amino acids are very rare, and some amino acids occur very frequently. They are very common in protein sequences. So the first thing they do is, in this block of amino acids, just count just count how many times an amino acid appears, okay? And divide this by the total number of amino acids. That's your frequency of amino acids. That's your Pi. So for each amino acid, again, I from 1 to 20 for all the amino acids. Maybe not all 20 amino acids may be available in this block of sequences. So you are not going to have any value for them. But you just count. For example, there are how many? Maybe 200 amino acids here. Out of these 200, maybe 40 of them are P, okay? The amino acid P. which is the first one, the first letter that occurs, and the first column, and the fourth column, and then the eighth column is all P's, okay? So it occurs, the P amino acid occurs a lot. So I didn't count actually, but let's assume out of these 200 amino acids, 40 of them were P. So this PP is going to be 40 divided by 200, okay, which is going to be 4/20. Now, let's get another amino acid, for example, L. L occurs, for example, 20 times, okay? I counted L, and I found that it occurs 20 times, and therefore, it's going to be 2/20. Okay, so the individual occurrence frequencies, the observed individual occurrence frequencies are PP and PL, 4/20, 2/20. Now, what is the probability to observe, since these are individual occurrence probabilities, the probability to observe these two together, the expected value of observing these two together is PP times PL. So you need to multiply 4/20 by 2/20. This is the expected number of observing P and L together. If you just randomly pick two amino acids from this, from your list of amino acids, if you just pick two of them randomly, the probability that one of them is, the first one is P and the second one is L, is going to be this probability, okay? This is the expected thing. Now, what we do is, okay, this is the expected based on the individual frequencies. This is what we expect by chance. Now, what we do is we actually, by going over each column, by analyzing each column, we actually count how many times P and L occur together, okay? That's your Qij value. Actually QPL, let's name that QPL for this specific example. You just, by going over all these columns, you count how many times, what is the actual, so this was the expected occurrence of P and L together by looking the individual frequencies. But what is the actual occurrence of these together in a column? You just simple counting how many times P and L occur together. And you do, this is your Qij, and the ratio of the observed occurrence to the expected occurrence of these two together is your likelihood ratio of mutation to P to L. And so, and you take the log of this value, so it means that you convert, so it's going to be a, either a positive or a negative, so the ratio here can be, since these are counts, they're all going to be positive. So to convert it to an integer, what we do is we take the log of this, this is called, therefore it's called logos ratio. If Qij is less than Pij, the log result is going to be negative, means that if the observed occurrence of P and L together is even less than the expected one, okay, they do not, in nature we do not observe this one mutated the other at all, so we should give it a negative score, okay? If Qij is less than Pi times Pj, this logos ratio is going to be negative. If it is greater, it's going to be a positive score, okay? So here's another example about two different amino acids, M and L. M occurs 1% and L occurs with 10% frequency. By random pairing, we expect 1 in 1,000 amino acid pairs to be ML. However, if the observed frequency of ML is actually 3 in 1,000, then the score of matching ML will be, and this log can be either in base 2 or base E. And these scoring matrices use the natural logarithm. And that's why we actually had in this formula this e to the minus lambda is due to the fact that the scoring matrices use log base e when computing the score. Okay, so your score would either be 1.5 or 1.1 based on whether you take logarithm base 2 or logarithm base e. Okay, and another thing that is done is these scoring matrices are usually provided as integer matrices, and these values are scaled by a constant factor to make them integers. So this 1.5 or 1.1 is just multiplied by some number to make all of them integers. So they are multiplied by a constant factor, and lambda that we use in the equation is the inverse of this original scaling factor. In other words, lambda can be found like this. So the original score is, so this, if the score between two amino acids is Sij in the scoring matrix, if the score is like 7, okay, this Sij is 7, this 7 is found by multiplying this logos ratio with 1 over lambda. So lambda is, the scaling factor is, for example, x, okay? Lambda is 1 over x, so therefore we multiply this logos ratio by 1 over lambda and obtain this integer score. So in other words, if you multiply your scoring value with this lambda, you are going to get the actual logos score. And if we take out the observed occurrence of Qij from this equation, this is what we are going to get. And what we know here is that these observed occurrences in these blocks of letters, they should add up to 1, okay? Because these are frequencies. I mean, if, for example, if you observed PL this many times, I mean, at the end, they need to sum up to 200, and the pairwise occurrences, they need to sum up to the whole observation, which is 1. So we do not know, since we do not have the exact alignments that they have used in these scoring matrices, we do not know these Qij's directly, but what we know is the fact that they need to sum up to 1. Your observations, all of your observations, these frequencies at the end, they need to be 1, these frequencies. So if we substitute this, if we substitute this Qij here, what we have at the end is this formula. And if, therefore, if we know the individual occurrence frequencies of individual amino acids in nature, and then we can, and if we also know the score values in a given scoring matrix, we can actually compute lambda. Lambda can be estimated by using this. So this is how they worked out to compute the E value of an alignment. Okay, this formula is the formula that is actually used in BLAST E value computation. X here is the bit score that they use. The alignment score is converted to an E value by using this formula, and you can actually inspect the result of a BLAST run, and at the end, they give the values of certain parameters at the end of a BLAST run in detail. You can find out what K is, what is the actual value of K, or what is lambda, what is the actual value of this lambda constant. You can look at a BLAST result to find this out. Okay, so this is how they compute lambda. Now, a Fasta, okay, this is how BLAST computes the E values. Fasta, on the other hand, uses something else. It uses empirical score distributions to compute the statistical significance. Now, therefore, they see that, okay, when they randomly align sequences, when they align randomly selected sequences and look at the scores. How do the scores look like when we align random sequences? And also, if we say that, okay, we just look at the maximum value of locally aligning a random string with a query string, And this, if we repeat this many times, and we plot a histogram of scores, we see that the resulting distribution is not normally distributed, or it's not, the actual distribution we are going to see is called an extreme value distribution, and it's going to look somewhat skewed to the right. So the green distribution here is a normal distribution, which is symmetric around the mean. And this Gumbel distribution shows that there are, since we are looking for the maximum value of the alignment, there are going to be more positive scores around, I mean, skewed to the right, meaning that there are more values which have higher scores. Even between random strings, we expect to get more positive values compared to smaller values, compared to negative values. And this mode is, the mode of this is like minus 0.5 or something. And this is the equation of extreme value distribution. You can plot by using, this is the function that will give you the extreme value distribution. And to extend the theory to gaps, it was really difficult. So they could not work out a closed form solution that can give the e-value of an alignment with gaps. And instead they use the e-value, the extreme value distribution to estimate the e-value. Okay, so the theory of local alignments with gaps is not well studied, as in without gaps. And what we have is mostly empirical results. We just run the alignments with random sequences and see how the distribution looks like. And BLAST therefore only allows a certain range of gap penalties, because of these previous empirical results only are studied only for certain gaps. And here at the end, this is the actual formula that BLAST uses. They use pre-computed lambda and k values for different scoring matrices. So for Blossom 62, they have a predefined lambda, and for different lengths, they pre-computed k values. And they first compute the row alignment score to a bit score using this formula, and they compute the e-value by multiplying this SSS is similar to that K, okay, it's the corrected score, so actually K is used here, and they use log 2 because the bit score is logarithm base 2, and here M is the query size and N is the database size, and L is the typical length of a maximal scoring alignment. So this is, but all these formulas, okay, these formulas are very similar to what we have done on match runs. They are derived from the same idea, okay. And what Fasta does is, it tries to estimate the probability distribution of alignments, similar to this, okay. It tries to estimate these distributions for every query. So for any query sequence, they collect a large collection of scores during the search of the database, and they estimate the parameters of the extreme value distribution based on the histogram of scores. So what is the parameter of extreme value distribution? It's just this X, okay? So extreme value distribution, this is extreme value distribution, but you can have it's a parametric distribution. Based on different X values, you can have different, you can even have a wider extreme value distribution or a narrower extreme value distribution. So they try to estimate the parameter X based on the histogram of scores. So it's completely an empirical way of computing the e-value. But the thing is, the main advantage is it provides reliable statistics for different parameters, even for different databases, different gap penalties. If you change, unlike BLAST, it doesn't use pre-computed things. To do these on the fly, it requires certain computation time. And this is the main difference between BLAST and Fasta. And the main purpose of these last two lectures was to give you an idea about how difficult statistical significance computation can be, but it's based on simple statistical foundations. We first studied a very simple thing, the coin tosses, and converted it to the match runs. And by detailedly analyzing it, studying it more, we can extend it to alignments with mismatch or alignments with gaps. And sometimes if we cannot do anything, the only thing if we cannot work out an equations, at the worst case, what we can do is we can try to plot the histogram of random alignments, how random alignments, their scores look like, and try to understand whether our score is much different than these random scores. How different our score is compared to these random scores. Okay, so if we cannot work out any formula, this is what we can do. Now, in the last 10 minutes before the break, I'm going to tell you another example, which is not related to a sequence alignment at all, but just to show you how statistical significance can be computed again empirically. This is what I did during my postdoc actually. This is when we were writing a paper. We had an algorithm to find clusters in graphs. So a graph is a discrete structure with nodes and edges, and graphs can be very large. You may have thousands of nodes and hundreds of thousands of edges between them. And given a graph, one problem in bioinformatics is this graph, for example, may show the relationships between proteins in cell, and your goal may be to find out certain subgraphs in this graph, okay, a subset of this graph in which the proteins are highly connected to each other. It's like you try to discover cliques, but not necessarily, or clique-like structures. Even given a very large graph, your goal is to find a subgraph in which the nodes in this subgraph are highly connected to each other. You are trying to find a highly connected component in this graph. So this is called cluster identification problem or graph clustering problem in general. And there are many, many different algorithms to solve this problem. And we proposed yet another clustering algorithm in this study. Now, however, the thing is, your algorithm produces a result. Says that, okay, I have found this cluster. Okay, I have found this cluster of nodes. These are the nodes, these are the edges. Now, we need to have some kind of a measure. How good is this cluster? Is this a really good cluster? Or maybe could you find this cluster by just randomly picking, for example, if you have found a cluster of size 6, okay, and there are connections between the nodes in this subgraph. If I just pick randomly 6 nodes from the graph, the cluster I found, is it going to be better than this random cluster or not? I mean, we need to measure how good this algorithm performs. Is it really finding the significant clusters, or we need some kind of a measure, or based on the graph. You could have, for example, if your graph, if your initial whole graph was itself a clique means that all the edges between the nodes in the graph exist. Okay, it's a complete graph. Okay, if it was a complete graph, you pick any subset, they're all going to be cliques. Okay, any subset would be a very highly connected graph. So if you run your algorithm on this, and when you say, okay, I found something really good, my algorithm works, you would be wrong, because any algorithm would work on that graph. So what we do here, the result we get should be based on the input graph. Even if, for example, the input graph may be very sparse, but if you're able to find very highly connected subgraphs, that means that your algorithm is working well. And the networks or the graphs in general, the biological graphs, the biological networks usually are very sparse. For example, even you have 6,000 nodes and you have 18,000 edges, okay? Even imagine you are given a graph with 6,000 nodes and 18,000 edges. Can you tell something about the sparsity of this graph? Is this graph very connected, or how connected is this graph? So what we can directly compute from these numbers is the average degree of a node. Which means that how many neighbors a given protein in this graph has on the average. It's 3, right? So if there are 18,000 edges in total and there are 6,000 nodes, on the average, if I just pick a random node in this graph, it's going to have 3 neighbors. What does this tell us? It means that the graph is very sparse, right? Because this 3 is a very small number compared to 6,000. Because you could this node could have 6,000 neighbors. Actually, 6,000 minus 1 neighbors if you do not count self edges. I mean, this node you'd select, there are 6,000 other guys around here. It could have been connected to all of them, but it's connected on the average to 3 of them only. So it means that your original graph is very sparse. Now, our goal here in this algorithm is to find a subset of nodes, okay? For example, to find 8 nodes in which all the 64 edges, actually, again, if the edges are undirected, it's going to be, okay, 8 times 8 divided by 2. It's going to be 28 edges. If this is 8 choose 2, okay? So if you are undirected edges, the total number of possible edges you can have in a node, in a subgraph of length 8 is going to be 28, right? Did I do the math right? I guess so. Okay, so it means that if you can find a subgraph in which you have 28 edges between these 8, no, I didn't do it right? So, oh yeah, it's true, yeah. So if you can find a subgraph in which all the edges between these nodes exist, it means that you find, even in this sparse graph, you have found something that is good. Now, but most of the time, you will not be finding, even if you are targeting these clicks in which all the edges are present between everything, okay? All the edges between them are present. Most of the time, such graphs, you will not be able to find such graphs, even if your goal is to find them. Sometimes some of these edges are going to be missing, but you are still trying to find these, if you still try to find it, even if a few of these edges are missing. Now, here is another question. In addition to trying to find out whether your algorithm is working good or not, we also sometimes need to compare, even within the same algorithm, 2 different results of different sizes. For example, here, do I have the, yeah, let's look at this. Yeah, just read the second bullet. A cluster of size 5 with 15 edges. Actually, we cannot have 15 undirected edges, but let's assume these edges they have certain weight. Or you find a cluster of size 20 nodes with 45 edges. Okay, which one is better? Now, when we compare these 2 things, we have a larger cluster. Finding larger clusters is a good thing if it's also highly connected. But when you're finding larger clusters, you are going to, your number of missing edges is going to likely to increase. If you add a new node to this, if you try to grow a given cluster, since your graph is a sparse graph, your likeliness to have edges to all the other nodes in this graph is going to decrease. So growing a cluster is not an easy thing. So you grow it and you do some sacrifices, your missing edges decreases, but which one is a better cluster? Is a large cluster of 45 edges is a good thing, or a smaller cluster but which is more condensed is a better cluster? So it's difficult to compare these two cases, right? I mean, if you don't have a single numerical measure for cluster goodness, you cannot measure this easily. Now, what we can do is we can compute statistical significance as a measure, as a universal measure to compare clusters of different sizes. So how are we going to do that? And the statistical significance is we are going to compute a p-value that's going to tell us this. If I select it, consider we have this cluster of size 5. If I just selected a random set of nodes with five nodes, could I achieve a similar clusters, the same number of edges? And we are going to ask the same question for the cluster of size 20. If I randomly selected 20 nodes, could I achieve this 45? And we are going to compare these two things with each other. And this statistical significance measure, so the p-value we compute is going to say what is the probability to find a random cluster of size n with the same number of edges. And here is what we do. We just select from the graph, from the given input graph, we select randomly 20 nodes. And we do this many times. How many times you need to do that? It's a question. I mean, 1,000 is 1,000 enough? 10,000 enough? Because you can select many so this number is very huge. 6,000 choose 20. Okay, this number is very huge. You can select many. There are many. I mean, it's a very large number that I cannot pronounce. There are many different ways to choose 20 nodes from a graph of size 6,000. But you choose, for example, what this shows that I think we did it like 1,000,000 times, because the count here shows 400,000. So we did it 1,000,000. For a million times, we selected 20 random nodes, and we counted how many edges this random cluster had. And most of them, and this x-axis, this is a histogram, this x-axis showed them 40% of this 400,000 of this 1,000,000 randomly chosen 20 subsets have no edges between them. Okay, they're not connected at all. And some of them, like 70,000 of them, like they have 4 edges, and 1,000 of them, or 10,000 of them have 7 edges. Now, this histogram tells me the likelihood of how does random look like. Okay, so this is how random looks like. And what you can do is, if your algorithm says that, I have found a cluster of size 20 which contains 5 edges. Okay, I have found a cluster of size 20 with 5 edges. Then you can immediately, by looking at this graph, you can immediately say that this is something expected, because there are there are 70,000 such random clusters with 5 edges in between them. But however, if you say that, if I found a cluster with 35 edges in between them, you say that now it's very unlikely, because I have very few or none. This is like 0 here all. So what we are going to do is we are going to normalize it, and this curve here looks like an exponential distribution. Okay, it's exponentially decreasing. What we can do is we can fit an exponential distribution to this curve, And this histogram is going to be a probability distribution. Then at the end, to compute the p-value, given a score, this can be computed easily if you have the formula for your probability distribution. What is the probability that x is going to be greater than or equal to your score? For example, 35. In this curve here, you can compute what is the, if you know the p here, your probability distribution, if you know that it's exponential, you can compute the probability of obtaining a score greater than or equal to 35 is the area under the curve to the right of this distribution. Okay? So the total area under this probability distribution is 1. The area to the right of 35 is going to be the probability of obtaining a score 35 or higher. So it's basically, if you know the exponential distribution, it's going to be you need to integrate this, I think, e to the lambda x dx. If I remember correctly, exponential distribution was e to the lambda x. So you need to get this integral from 35 to infinity, and this is going to give your probability. And this is what we did. Yeah, actually the exponential distribution is lambda e to the lambda x. The integral is going to drop the lambda. So this is our exponential distribution. And there's only one, this is a parametric distribution. The only parameter is lambda. And for exponential distribution, it's the rate of the fall, you can consider it as the falling rate. And this figure shows how different lambda gives you different exponentially distributed variables. And our goal is to compute this. What is the probability of obtaining a score W or higher? And the integral turns out to be, okay, this infinity seems like a difficult thing here. How are we going to do that? It turns out that part is going to be 0. E to the minus infinity is going to be 0, so you're just going to drop that out. So at the end, this integral is going to just be e to the minus lambda x. Okay? This is the area under the curve is equal to the e to the minus lambda x. X is your score here. Now, based on what this slide shows is that clusters of different sizes, size 5, size 10, size 20, or size 30, have different looking exponential distribution. So the lambda parameter in these distributions for different cluster sizes are different. So you need to compute these lambdas for different cluster sizes in order to be able to compute the p-value for a given cluster size. And here is the example. If lambda 5 is 1.7 and if lambda 20 is 0.36, okay, for different cluster sizes, and suppose we have found a cluster of size 5 with 15 edges and 20 with 45 edges, if we compute the p-values, we see that the cluster of size 5 actually is more significant. It's mean the p-value is smaller. It's more significant than the cluster of size 20. Okay, so if you say which cluster is a better cluster, you would actually choose the cluster of size 5 because it's less likely to find such a cluster by chance. Okay, and that would be your main justification. If you say, why did you choose cluster of size 5, not the size 20, you can argue, okay, the probability that to get a cluster of size 5 with edges some 15 is less likely. Okay, this probability is like 10,000 times smaller than the other probability. So it's 10,000 times less likely to find this cluster of size 5 with those edges. Okay, so this is how you compute p-values, and this is the computation of statistical significance is very common actually in many different fields of science. So what my suggestion is that whatever you'll have learned here, do not just use it in bioinformatics problems. You can also use the same idea for different algorithms in different domains. The the same approach should be used to have some kind of a scientific meaning of your results. Okay, so let's have a 10-minute break, and we are going to continue with the new topic.